Hello, my name is Amanda Scharenbrock, and I work in Dr. Misha Peronsky's lab at the University of Wisconsin-Madison. Today, I will tell you about the effects of anesthetics on mitochondria mutants in a fruit fly model. Animals with mutations in complex 1 of the mitochondrial electron transport chain, METC, exhibit behavioral hypersensitivity to volatile general anesthetics, VGAs, and may be susceptible to VGA-induced deleterious collateral effects. We previously found that a nuclearly encoded complex 1 gene, ND23 in fruit flies, results in a hypersensitivity to VGAs. ND23 is the ortholog of mammalian NDUFS8. Here you can see that ND23 NDUFS8 is located in the matrix part of complex 1. We found that exposing ND23 mutant flies to isoflurane resulted in lethality. Isoflurane toxicity was exacerbated in hyperoxia. However, sevoflurane did not result in toxicity under the same circumstances. We were curious whether a different mutation in complex 1 would have a similar phenotype. We obtained mutants in the mitochondrially encoded ND2 subunit, which is located in the part of complex 1 that is in the inner membrane of the mitochondria. I compared the ND23 and ND2 mutants in isoflurane and sevoflurane, as well as normoxic and hyperoxic conditions, to determine to what extent the mutants were susceptible to anesthetic toxicity. We administered anesthetics using the serial anesthesia array, or SAA. This SAA consists of agent-specific vaporizers, flow meters, and a serial array of anesthetizing positions. Flies are put into the conical tubes and placed on the array for a specified amount of time. Here's more information on the mutants, specifically the lifespan curves being shown here. The ND23 mutant was characterized by the Ganetsky lab at UW-Madison. In the homozygous form of the mutant, it has a greatly decreased lifespan, living just past 20 days old. The mutant also shows neurodegeneration. As for the ND2 mutant, it exhibits a similar shortened lifespan, once again dying just after 20 days. Prior to the experiment, we maintained the flies at 25 degrees Celsius in bottles. On day 0, we clear the bottle and allow new flies to hatch for 2 days. These young adult flies are then transferred to 29 degrees Celsius and aged until they are 11 to 13 days old. These flies are then put on the SAA for 2 hours under the specified condition. 24 hours later, we checked for mortality, which is shown here. First, we have the ND23, in which you can see a mortality phenotype in isoflurane in normoxia. This response greatly increased in hyperoxic conditions. However, there is no mortality with sevoflurane. Moving on to the ND2s, you can see a similar effect with isoflurane in both conditions. A similar reaction also holds with sevoflurane in normoxia. However, by adding hyperoxic conditions to sevoflurane, mortality increases. These data lead to several conclusions. Mutations in both nuclearly and mitochondrially encoded subunits of the METC complex 1 increases susceptibility to isoflurane toxicity in flies. The mutants differ in their susceptibility to sevoflurane toxicity in hyperoxia. Mutations in different complex 1 subunits confer VGA-specific susceptibility to toxicity. Moving forward, the genetic approaches available in flies will allow us to understand the risk of VGA toxicity in carriers of mitochondrial mutations. My thanks to the Peronsky lab, Wasserman lab, and ISAP for this opportunity.